model is divided into two phases, interphase and mitosis. Interphase includes the three big green, yellow, and red parts. There are different parts in each phase. Alright, let's start with interphase. G1. Take this cell and watch what happens as it goes through the G1 stage. Let's see why that occurred. G1 contains the growth of the cell. G1 stands for gap 1. It is one of the longer processes in the cycle, which explains why it has a bigger piece of the chart. All the cell does is grow, grow, and grow until it meets a certain standard which is measured at the G1 checkpoint. Synthesis. DNA replication is a semi-conservative process, in which means that one strand of the parent double helix is conserved. Polymerase are the building blocks of DNA, and add nucleotides to the DNA strand. As you can see here, a helicase is essentially what unzips the DNA from the double helix. GAP2. G2 contains more growth, gets the cells ready to divide. G2 equals GAP2. It allows cells to check if the DNA replication process was correct. G2 is also the phase where organelles start to replicate as well. GAP2 is an entry into mitosis. Mitosis. Mitosis contains a phrase called PMAT. P stands for prophase. This is where DNA starts winding into chromatids. These are held together by a centromere. This is called a chromosome. During metaphase, the chromosomes line up in the middle of the cell. This brings us to anaphase, where the chromosomes are pulled apart with the spindle fibers on each side of the cell. Telophase includes chromosomes that unfold to become a chromatin, while the spindle fibers also break down, and new nuclear membranes form. Cytokinesis. The cyto in cytokinesis refers to the action of splitting the cytoplasm completely to form a daughter cell. After the cell is done splitting, the cell goes on for its daily tasks in gap 0 or keeps repeating the cell cycle.